Hey guys, we are back, and today we're going to talk about biotechnology, specifically in the areas of forensics and medicine. So, remember that with this, we are looking at Georgia Standard of Excellence SB2, which is all about how genetic information is expressed in cells. And we're focusing on Substandard C, which is looking at the use and ethical considerations of biotechnology in forensics medicine and agriculture. So, we are going to talk about advancements in technology relating to both economics and society. And these advancements could include GMOs or genetically modified organisms. So, by the end of this session, you should be able to discuss in detail the ethical considerations of biotechnology in forensics, medicine, and agriculture. So, let's look at biotech in medicine.
At the dawn of medical biotechnology, a new frontier in healthcare emerged, defined by bold experiments and revolutionary discoveries. This era commenced with pioneering work in biotechnology as scientists started to decode the complex biology of life. Genetic engineering emerged as a groundbreaking achievement, allowing for the manipulation of life's blueprint. It paved the way for biotech drugs, including the production of insulin through recombinant DNA technology, a beacon of hope, demonstrating the journey from lab bench to patient bedside. From these beginnings, biotechnology has grown, offering potential solutions to some of medicine's most challenging issues, including gene editing and personalized treatments. This foundational work set the stage for an exciting future where our ambitions could reach as far as our ability to explore. Continuing our exploration through medical biotechnology, we focus on CRISPR-Cas9 gene editing and regenerative medicine, heralding new healthcare paradigms. CRISPR-Cas9 offers precision in correcting genetic disorders, embodying the hope for treating previously incurable diseases. Beyond gene editing, regenerative medicine through stem cell therapy and tissue engineering opens possibilities for repairing damaged tissues and organs, such as heart tissue after a heart attack. These advancements are transforming treatment approaches, underlining human ingenuity and a dedication to improving lives. They serve as a cornerstone for personalized medicine, tailoring treatments to individual genetic makeups and underscore the unfolding journey of biotechnology in healthcare. We now enter the era of personalized medicine, a transformation underpinned by genomic sequencing, targeted therapy, and predictive diagnostics. This approach leverages an individual's genetic information to make treatments more effective and efficient. Genomic sequencing offers a disease susceptibility and therapeutic roadmap, while targeted therapy designs treatments that minimize harm to healthy cells. Predictive diagnostics provide tools for optimized treatment strategies. Together, these innovations signal a shift towards healthcare that is deeply personalized, setting the stage for advancements that promise enhanced care and open doors to future possibilities in medical biotechnology. As we delve deeper into the landscape shaped by personalized medicine, we encounter the domain of biopharmaceuticals. Monoclonal antibodies, targeting specific antigens with unparalleled precision, and innovative vaccine developments are at the forefront, improving public health protection against formidable pathogens. Biosimilars play a critical role, making life-saving treatments more accessible. These advances embody a new chapter in medicine, where each development brings us closer to conquering complex diseases. In the realm of biopharmaceuticals, we're not just witnessing progress, we're part of a movement towards a future filled with healing and hope. In the concluding chapter of our narrative, we envision a future redefined by medical biotechnology. This journey has illuminated the path from gene editing and regenerative medicine to the advent of personalized medicine and biopharmaceuticals, setting the stage for a new era. Yet, as we embrace these advances, we're also tasked with ethical considerations, ensuring equity in innovation. The true promise of medical biotechnology lies in its potential to transform lives globally, fostering a collective approach to overcome health challenges. As we look forward, we do so with optimism, guided by the choices we make today. For healthcare professionals, enthusiasts, and students, the future of healthcare is not only unfolding, you are actively shaping it. Together, let's commit to a healthier future.
All right, guys. So, one of the biggest areas where biotech and medicine is important is insulin. So, production of insulin is a huge, important part of everybody's body. And there are some people who have diabetes, they are type 1 diabetics. Their body does not make insulin. And that is a huge problem because insulin is needed to balance out glucose. And so, what scientists have discovered is that they can actually create what's called a recombinant plasmid or recombinant DNA. And in doing so, they can take the insulin gene and insert it into bacteria. Remember, bacteria reproduces super, super, super fast. Because bacteria reproduces super fast, they can produce lots of that particular gene, insulin, and make lots of insulin that they can then extract that can then be sold to patients who need to take insulin. So here is an Amoeba Sisters video telling us about how to make insulin.
Or techniques. Let's focus more on the whole bacteria producing insulin example to illustrate how this was done. Focusing first on some basics that we know, here is a human cell. Like most cells in your body, it contains a nucleus. And like most body cells, that nucleus contains the organism's entire DNA code. With some exceptions, each body cell you have contains all of your DNA. If you recall, genes are made of DNA, and so there's a gene that codes for making the protein insulin in most of your body cells. While this gene could be removed from a cell's DNA, the gene for insulin can also be synthesized in a lab. This insulin gene can be inserted into a bacterial plasmid. A plasmid is like an extra set of genes, in addition to the bacterial chromosome, that bacteria can use. Plasmids tend to be in a circular shape. Plasmids are common in bacteria. You can also find them in yeast cells. But to get specific DNA into the plasmid, you have to make space for that. For that, you can use restriction enzymes. Restriction enzymes are enzymes that cut in specific spots, like teeny tiny scissors, and you can cut a specific spot in the plasmid so you can add in that human insulin gene. Ligase, you remember ligase from our DNA replication video, can be used to help seal it into place. This is now considered recombinant DNA because it contains not only the plasmid DNA, but also the DNA of interest, the gene for producing insulin. The recombinant DNA is made up of DNA from different sources. In order to encourage a bacterium to pick up the plasmid in transformation, certain chemicals and temperature changes may be used. Once it picks up the plasmid, when the bacterium reproduces by splitting, the resulting cells will both inherit the plasmid, and then their daughter cells will inherit it, and theirs. You get the picture. In this way, the plasmid continues to be produced over and over. The bacteria can use the human insulin gene to produce human insulin, and the insulin can be purified in a lab setting to be used for humans.
All right. So, if you know anybody who is type 1 diabetic, that's how their insulin is made in order to ensure that they are able to survive. So, this is called recombinant DNA, and it literally just means it's a combination of two DNA strands that have been constructed artificially. And so, genetic scientists can do this to create a unique DNA strand for many different purposes. They can use lots of different types of techniques, but one of the biggest examples of this is to make insulin. And like naturally occurring DNA, recombinant DNA has the ability to produce proteins from that recombinant section of DNA, which is why it's able to produce the insulin. So let's think about, what do you remember about DNA? Take a minute, process. So is DNA single-stranded or double-stranded? Going to give you a couple of questions. That's going to be one of them. So we know that DNA is a double-stranded molecule. It's in the shape of a helix or a twisted ladder, kind of like a spiral staircase. It consists of four nucleotides. Remember that in Club DNA, adenine is not cheating yet, so it's adenine, thymine, cytosine, and guanine. It's found in the nucleus of eukaryotic cells. In prokaryotic cells, remember, they don't have organelles, so it's found in the cytoplasm. It's got kind of a region that it's located in, but it is in the cytoplasm. And it is the blueprint for building proteins. It is super important to us. And every individual organism has a unique set of DNA. Now, there are lots of similarities from one organism to the next, but everybody's DNA is slightly unique to them. Now, with DNA, we can actually solve crimes, which is pretty wild to think about, that you can use the DNA that might be left behind at a crime scene in order to solve a crime. But let's see what the Amoeba sisters have to say about it.
Gel electrophoresis can be used to separate molecules based on how big they are, their size, and it's especially useful with DNA. Let's look at DNA real quick. So here is a guppy cell, here's the nucleus in the guppy cell, here's the DNA in the nucleus of the guppy cell, and if you were to zoom into the DNA, here is a nucleotide, which is a building block of DNA. See those phosphates in the nucleotides? They're a bit negative. Well, they contribute a negative charge anyway to the DNA. So if we look at this whole DNA here, it gives that DNA a negative charge. That's a big deal because gel electrophoresis, which again separates molecules based on size, relies on the fact that DNA molecules have a negative charge. Okay, here's a gel electrophoresis machine. The point of the machine is to be able to have an electrical charge running through a gel. So here's the gel, typically made of agarose. Agarose is a polysaccharide polymer, which if you remember from our biomolecule video, polysaccharides are carbohydrates. Yeah, usually agarose comes from seaweed. The agarose gel itself lets the DNA molecules travel within it. One end of the gel has these holes called wells. The wells are where the DNA is placed into. The area of the gel where the wells are is negatively charged, and the area of the gel here is positively charged. So guess where the DNA will travel towards? Well, since it's negatively charged, it's going to travel to the positive side. So typically when you're analyzing DNA in electrophoresis, you use these restriction enzymes to cut the DNA up into tiny pieces. Restriction enzymes have the ability to cut up DNA in very specific areas, often related to the specific DNA bases, making restriction enzymes very useful in biotechnology. So if I had baby guppy DNA and adult mother guppy DNA, and I want to compare them, then I would want to use the same types of restriction enzymes in both DNA samples. If I used the same type of restriction enzyme, it should be cutting the DNA at the same identification points in the DNA samples. However, unless the mother and baby guppy are clones, and they're not, those pieces that result after the restriction enzyme is done with them may be differently sized because the DNA of the baby and mother guppy had some differences in the sequence of their DNA bases. So the DNA samples both are cut up into multiple pieces by the same type of restriction enzyme, and then those samples are loaded into the gel. Sample 1 and sample 2. If we turn on the machine and let the DNA run through the gel, the DNA moves towards the positive side. But some pieces of that cut up DNA will move faster or slower than other pieces. Longer DNA pieces tend to have a higher molecular weight, and they take more time to make it across the gel when you compare it to shorter DNA pieces which move at a faster rate. So what you end up with is that these DNA fragments spread out, with the longer pieces closer to the wells and the shorter pieces closer to this opposite side of the gel. These are called DNA bands, but to see them, you usually need to stain the gel itself and view it under a UV light. Now let's compare the DNA bands in this hypothetical, simplified guppy situation. The bands aren't going to be identical because these fish are not clones, but I can compare how similar the bands are and compare that to other mother guppy samples to look for relationships. Let's say that we have 3 mother guppy samples to view, and these are the only possible mothers from the fish tank. Which one appears to be the most related to the offspring in this case, and has a high likelihood of being the mother? Well, this one. But we can't be 100% sure with this. It would be helpful for me to know the father guppy sample too, because this will give me more insight. But if these are the only fish in the tank, it's a very high likelihood with this case.
All right, guys. So, DNA fingerprinting is what they were basically just talking about. That's how they can look at the DNA bands and they can figure out if the DNA matches with either a parent or if the DNA matches a crime scene sample. So it was in 1984 that a British scientist, Sir Alec Jeffreys, actually pioneered this method and this technique of DNA fingerprinting, which allows for scientists to be able to analyze an individual's unique DNA pattern, which then can be used for identification processes or purposes, I'm sorry. And this breakthrough has revolutionized forensic investigations and has been used to solve countless mysteries. Without this, we would have a significant trouble or significant problem with solving different unsolved crimes. So the Amoeba sisters just did a great explanation, but this process is called gel electrophoresis. You have to extract the DNA, you have to amplify it through something called PCR or polymerase chain reaction, and then you have to analyze it using that gel electrophoresis. And what you're looking for is matches. Who has the most in common? So resulting DNA patterns are always going to be unique to every individual, except for identical twins, because identical twins share the same DNA profile because they come from a single egg and a single sperm that join together into a zygote. And then at some point during the very early stages of cell division, that zygote split into two separate individuals. Because of that, they have the exact same DNA. Otherwise, though, if they are not identical twins, then you are going to see differences to those patterns, and you'll be able to match up those patterns to figure out who the parent might be or who the murderer might be. And you can see here, we've got a crime scene DNA sample. We've got a suspect 1, 2, and 3, and you can look at what they have the most in common. And I would say from here, what do you guys think? Take a minute, guess. I would say suspect 2 is our suspect. So you can also use this for paternity testing. You can use it to determine biological parentage. You can use it for genealogy. You can trace ancestry. You can identify relatives. You can use it for conservation biology. So you can use this to study populations and track a variety of different endangered species. And then we have cloning. So I've got a National Geographic article that we're going to add into our Canvas classes if you're interested in learning more about this. But cloning is just the process of creating identical copies of a living organism. Nearly 30 years ago now, Dolly the sheep became the first cloned mammal, which sparked major debates and lots of excitement. So was this process ethical? Should we have done this? So if you're interested in getting more from that, feel free to grab that National Geographic article. I'm going to add it into my resources in Canvas. But let's see what this video tells us about cloning.
Can you spot the two clones? When we talk about clones in science, we mean organisms that are identical copies. They have the same DNA as each other. Identical twins are examples of naturally occurring clones. Both plants and animals can be cloned. Let's start with plants. A gardener, farmer, or plant breeder might want to make many copies of a particular plant quickly. The easiest and cheapest way to do this is to take cuttings. Shoots are cut from the parent plant, then the end of each shoot is dipped in hormone rooting powder and placed into a pot of soil. The hormone rooting powder will encourage the cutting to start growing roots, and soon a whole new plant will have grown which is identical to the original plant. The method works because the shoots of the plant contain stem cells, which are able to differentiate to form different cells and tissues. You can find out more about this process by watching this video. However, you might want to produce hundreds of plants from only a small piece of plant tissue. To do this, another method called tissue culture, also known as micropropagation, is used. Here are a few cells taken from the parent plant and placed onto a nutrient jelly using antiseptic technique. This means making sure no microorganisms contaminate the jelly. The cells will start to differentiate and form new plants. Animals can also be cloned, but different techniques need to be used. In animals, only embryo stem cells have the capability to differentiate all the different types of cells found in an adult. There are two methods you can use. The first is called embryo cloning. A farmer might have a cow that gives a lot of milk and wants to use her to create many calves. The cow is artificially inseminated using the sperm of a boar. The embryos grow until they form a ball of embryonic stem cells. Before they become specialized, the embryos are removed from the uterus, divided up into separate embryos, which will all have the same DNA, and then each is placed into the womb of a different cow. These cows are surrogate cows. They are just being used to grow the embryos until they are born. The calves will be clones of each other, but not a clone of the mother due to the boar's sperm. Hopefully, the calves will have the characteristics that the farmer desires, but as sexual reproduction is involved and the genes from both parents are randomly mixed, there is always the chance that they won't. There is a way to get around that problem, and that is to use another technique called adult cell cloning. This is used to form a clone of an adult animal. You might have heard of Dolly the sheep. She was the first mammal to be cloned back in 1996. There are several steps to this process. An unfertilized egg cell is taken from an adult female, and the nucleus is removed. A body cell, such as a skin cell, is taken from a different adult. The nucleus is removed from this adult body cell and is inserted into the egg cell. An electric shock stimulates the egg cell to divide to form an embryo. These embryo cells contain the same genetic information as the adult body cell. When the embryo has developed into a ball of cells, it is inserted into the womb of an adult female surrogate to continue its development. So 3 different adults are used. The baby born will be a clone of the adult that donated the body cell because it has the same DNA. This technology is very controversial and raises many ethical issues. One worry is that it could be used to clone humans. Questions raised by this might be, how many embryos would be destroyed in order to successfully clone a person, and would the clone have the same human rights as others? So in this video we have looked at what a clone is, and the different methods used to clone plants and animals.
If you liked the video, give it a thumbs up and don't forget to subscribe. Comment below if you have any questions.
All right, you guys. That is everything that I have for you about genetically modified organisms and about biotechnology in medicine and in agriculture and in forensics. So we will talk about this more in class. I hope you guys have an awesome rest of your day, and I will see you soon. Bye, guys.